So when the samples come back to the lab, the first place they come to is this room, which is the sample check-in room. So this is where we inspect each sample visually to make sure that it looks as we would expect it to. There's a whole range of sort of quality control checks that are gone through, and all of the information about the sample is recorded so we can make sure that it's going to go through the right analysis workflow through the lab. Once they've been checked in, they're ready to go into the next part of the process. So our laboratories are set up with a lot of different individual separate rooms, and that's because our samples are very vulnerable to contamination because we're working with just tiny, tiny amounts of DNA, and that's especially true for the water samples. So we have to have different labs where we extract DNA from different types of samples. Molecular biology is this amazing process where you take tiny amounts of colourless liquid, mix them with other tiny amounts of colourless liquid, heat them up, cool them down, spin them quite a lot, and out at the end pops a whole list of species. So once the DNA has been extracted and purified from the samples, the next stage that it comes is into this room, which is the PCR setup room. This is an absolutely critical part of our process. It's where we make millions of copies of the bits of DNA that come only from the taxonomic group that we're interested in. So if we're interested in doing a fish analysis from our DNA, for example, we set up a reaction which makes millions of copies of just the fish DNA so that we can sequence that later on and get a good clean data set that's really powerful for showing fish. The next stage is to come into this lab, and this is where that amplification reaction happens. A small machine which goes through many cycles of heating and cooling the tiny amounts of colourless liquid that we're working with. Every time you go through a cycle of heating and cooling, it doubles the amount of copies of the DNA from the taxonomic group that we're interested in. And so you get this sort of exponential increase in the amount of DNA. So that amplification reaction takes 1 to 2 hours. It gives us lots of DNA to be able to sequence. So this is our sequencing room. These are the sequencing machines. We'll run about 100 samples together to turn the amplified DNA into a list of DNA sequences. It takes about 2 days for each one to run, and the data is automatically sent to our server ready for analysis by our bioinformaticians, who take that massive text file of A's, T's, C's and G's, clean, sort, separate those sequences, match them against reference databases so we can turn it into a species by sample table that's ready for ecological analysis.